So in this video we're going to have a think about what the cell actually does most of the time. And if you have a look at this graphic, you will notice that most of the time the cell is in interphase. So you can see that most of the time it spends its time in interphase. It's only actually doing division for a really short amount of time. And when it is doing division, most of that time is spent in prophase. So it takes a while for the chromosomes to condense and for them to start organizing themselves in the right place in the cell. And as soon as they hit the equator during metaphase, they're pulled apart in anaphase, and then the cell starts to reform its nuclear membrane, and then the cell divides itself into its cytoplasm in two, and then it's ready to get on with its job again. So we call this whole process the cell cycle, and that's what we're going to have a look at in this video and why the cell cycle is important. So here is the cell cycle, and at the top here we have mitosis. This is cell division, and you can see that the cell is dividing, and the two identical cells that get produced go off into their own version of the cell cycle. So we'll follow this cell around the cell cycle. Here it is in interphase, so all of this bit around here is interphase, so starting here, all the way around here, all the way up to mitosis again, that's all interphase. So when the cell starts at interphase, it is usually pretty small because it's just been divided in two. So its first job in G1 is to grow. And you'll notice the cell has got bigger. Now, if it gets big enough, there's a little bit of a checkpoint here, it will then multiply its DNA. So it will replicate its DNA. You'll see that the chromosome here is made up just of one strand, and now it's made up of two strands, joined together by a centromere. If that is successful, the cell will then grow its organelles, and there will be a checkpoint here, making sure it's got enough organelles to split into two new cells, and then it will go through mitosis. So that is the cell cycle. Let's have a look at what's happening to the amount of DNA that's going in the cell. At the beginning, G1, the cell is increasing in size. Then S phase, the cell is replicating its DNA. So you can see the amount of DNA is increasing and it doubles. So every chromosome makes an exact copy of itself and holds it next to itself, attached by the centromere. Then in G2, that's when the organelles are being made to make sure there's going to be enough for both of the cells. And then the cell actually undergoes mitosis. And at the very end of mitosis, when the cell is splitting in two, the two new cells go back to having the original amount of DNA. Because of course, during mitosis, you produce the identical cells at the end of the process. Now, this is really important to understand. Great. It's mostly important for knowing why and how things go wrong. And the main thing that can happen when it goes wrong is cancer. So I was emphasizing earlier that there were checkpoints. There is a checkpoint, very important checkpoint here at the end of the first growth phase. It has to pass that checkpoint before you can manufacture DNA. And there's a very important protein that's involved in that called P53. P53 is known as a tumor suppressor gene, and P53 has to be carefully regulated at that point. It's a checkpoint there, and if P53 determines that things are not right, it will stop the cell cycle happening because you do not want there to be problems and the cell cycle to continue. So P53 will prevent tumours growing. And if everything is right, P53 will be removed and the cell cycle can happen. if something goes wrong with P53 and DNA is made and the cell cycle goes on and on without a check, then you end up with the cells proliferating, and that's the first stage of a cancerous growth. If there are further mutations within that clump of cells, you might actually end up with a cancer. Now, if P53 is not working, it doesn't mean you definitely have cancer, but you might end up with some cells proliferating. But of course, there are other checkpoints. So check out up here, there's a checkpoint with other important genes that make sure that the DNA is undamaged, that there are enough organelles. As long as that checkpoint is met, then you can go through mitosis. And even during mitosis, actually during metaphase, there's a check again to make sure all the chromosomes are attached to the spindle in the right way. And if they're not, the cell stops there and the cell won't be allowed to continue through metaphase. So the cell cycle is very carefully regulated to make sure that cancer can't occur. And when cancer does occur, it's usually because one of these checkpoints has failed, and actually probably more than one of these checkpoints have failed. So this will help us to link into other modules later on in the year.